The following case study presents the research status, the gap analysis, and a 6-month strategy proposal for Barth Syndrome Foundation that wants to emphasize its support to drug development. First, I gathered the clinical features of Barth syndrome. It's not my intention to explain the syndrome you work with or live with every day, but rather to expose the main issues in Barth syndrome from the patient point of view. After talking to your patients and listening to their testimony at conferences and reviews, it appears to me that the main problems were the cardiomyopathy, the fatigability and exercise intolerance, the neutropenia, and the 3-mg aciduria. When we ask teenagers or young adults what blocked them in their life, they answered heart failure, fatigue, and fear of infection. What I see as a consequence of this tableau is first the challenge for diagnostic and research due to phenotypic diversity, and second, outside of a medical point of view, it's the social aspect of patient alienation. So what has been done up to now for Barth syndrome in terms of academic research? In the center is a map of the federal funding. What is very positive is the effort made for the development of various models, from cellular to animal models. You can see that the mainstream of research has been focused on one of the consequences of the TAS mutation, the cardiolipin dysfunction, and surprisingly, very little has been done on the function itself of the protein of interest, tafazine. Some studies have been also performed on the more general function of mitochondria and on lipids, but really too few on neutropenia and hematopoietic differentiation. In Europe, the research is performed in a different optic, focusing on stem cells in cardiomyopathy, neutropenia, the tafazine involvements in cell differentiation, and most importantly, cancer. The research in China has similar focus as Europe, with the tafazine in cell differentiation and cancer development, as well as cardiomyopathy. Your foundation has changed orientation, putting efforts in gene therapy and starting studies on genotype-phenotype study, as well as neutropenia mechanisms, but still keeping developing models and fatty acid oxidation. That is the global status for research. Now, where are the gaps in research? Gaps in research depend on what you want to obtain, knowledge on the disease, or where and how to act in order to find a cure. We know that the TAS mutation means no functional tafazine that triggers impaired ratio of ml cardiolipin. Well, it's not that simple. Here you see the representation of a cell with the nucleus, where the TAS gene is in the DNA. It goes to the endoplasmic reticulum for transcription and then translation into the protein tafazine. Tafazine has an address that should take it to the mitochondria. Most of the research, as we saw before, has been done on cardiolipin or the mitochondria. But the consequence of the mutation does not stop there. We know that the cytoskeleton, what gives its shape to a cell, is altered in Barth syndrome cells patients. And it has been also shown recently that proteins that are in the rest of the cells are also modified. Through the techniques of proteomics that you might have heard before, they saw that 91 proteins that are not at a normal level in Barth syndrome. Each of these proteins have a function. A protein sometimes needs to be activated in order to exert its function. This state of activation depends on its level of phosphorylation, which means that the protein has a phosphate group attached or not. The general state of phosphorylation is what we call the phosphokinome. A very recent study has shown that the phosphokinome in Barth syndrome is altered in many different functions of the cell. This kind of research will reveal new pathways triggered by the lack of tafazine, and then new targets for drug development to get away from this highly specialized approach and lack of investment in therapies. To summarize the opportunity in Barth syndrome target identification, imagine the TAS mutation as a big rock falling from a cliff. You can stop the rock, this would be the gene or mRNA therapy. Or you can let the rock fall and try to stop the other rock it's going to bring along. That would be the downstream signaling pathway, cardiolipin, phosphokinones. What we have now is only symptomatic therapies, improving cardiac efficacy, diuretics, ACE inhibitors, antibiotics, trying to restore the damage of the disease. The action points for Barth syndrome can be gene therapy, going further with the AAV9, the drug repurposing. You already have the ELAM from state therapeutic in clinical trials, but you could increase your participation and join other clinical trials that might benefit your patients, like contact other biotechs like Neurovive for the KL1333 or Reneo Pharma for the REN001 or other biotech companies. For the new targets, the new studies showed a variety of interesting targets and downstream pathways that are of high interest to investigate. I listed here the most relevant to begin with. The phenotypic screening should be one of your main focus, with getting the most valuable chemical libraries from pharmaceutical industry and biotechs.